Perfect. Great. Thank you first so much, Noah and colleagues, for organizing this great event today. I'm now very excited to conclude with the talks here. I will cover what theoretical population biology contributed, years and decades of theoretical population biology research in the response to the COVID-19 pandemic. And in that, we'll go through three different topics. So we'll touch upon the reproductive number, a concept which has been developed for decades, cover logistic growth, both with applications to the COVID pandemic, and then look at where are we now and what does the future promise. All right, so reproductive number. Here you see a sketch of a healthy population, and assume at some stage somebody gets sick, a first infection. So be it a first infected individual back in Wuhan in 2019. And now further assume this person infects on average 2 people. So we have then 3 people in total infected. Those 2 people on average may infect again another 2 people. Makes 4. Those 4 infect another, each infect 2, makes 8, and so on. And so this 2 is actually what I had here now exemplary, the reproductive number of this hypothetical outbreak. The reproductive number is defined as the expected number of secondary infections caused by a single infected individual. And this quantity is central in epidemiology. And actually, there have been many decades of work around this reproductive number. And I would claim pretty much throughout the 20th century, there were in different fields, demography, population biology, epidemiology, concepts not necessarily coined as the reproductive number, but concepts discussed. And actually, I think rather than giving names here, it would be a huge list who contributed to it. I recommend this article by Hans Hesterbeck from 20 years back, A Brief History of the Reproductive Number and a Recipe for its Calculation, where he really outlines how the whole 20th century was influenced or work contributed to how we now use the reproductive number. And in fact, most of the names which were dropped in Mark's talk earlier with chapters of this book he laid out, those names also in some way were involved in this reproductive number. So what is it? Why do we care in epidemiology, and what does it mean? Well, first, if R, the reproductive number, is bigger than 1, it means the epidemic is growing exponentially. One person on average infects more than 1 person, so has more than 1 children, meaning things spread. While if R is smaller than 1, the epidemic declines. So, well, one could argue we can just look and see if case numbers go up or down. Why do we care about this reproductive number? Well, one first thing is, and here we do an additional definition, the reproductive number at the start of an epidemic, called also R0, it is how fast is the pathogen being transmitted, so what's the expected number of secondary infections at the start without people having any immunity nor any measures were there. Meaning that was in 2019 in Wuhan, when nobody even knew about SARS-CoV-2, or then in the early 2020 in most of our countries when the virus had already entered, but we didn't really realize. And so the virus just spread with our normal lifestyle, and everybody was susceptible to the virus. So then it became a big question, what's this R0? And in general, there's a lot of scientific work on how to calculate R0. Why do we want to do that? Well, if we know R0 when an outbreak starts, then under simple population dynamic models, we can derive that the epidemic is only starting to decline again once 1 minus 1 divided by R0 of the population obtained immunity, be it through vaccination or infection. So people are immune to being infected by the disease. Why this term? So I had this example of 2. 1 person on average infects 2 people. Now, If we plug 2 in here, we get a 0.5. And you can now envision if I, on average, infect 2 people, but half the population is already immune, well, then I only find 1/2 who is susceptible, meaning rather than infecting 2 people, I only infect 1. The other one I would have infected is already immune. So once half of the population has immunity with an R0 of 2, the epidemic starts declining. And early in the pandemic, this was, of course, it was very important to know what is this R0, because that will give us a sense of when do we think case numbers will go down again if we weren't to do anything. And so in what follows, I actually want to highlight a few examples where first this R0, but then throughout the pandemic, the reproductive number played a role. So first, early on, so very early 2020, it was unclear what is the R0. We just saw in Wuhan increasing case numbers. And Rio and Althaus were one of the first to actually quantify R0 for Wuhan. What essentially they did is they looked at the case numbers and assumed that roughly every 5 days, that's from contact tracing was suspected, every 5 days, on average, a person infects and transmits the disease. An infected individual infects disease after 5 days. Then at day 5, those people are getting newly infected again after 5 days, in fact. And roughly every 5 days, this epidemic was doubling, meaning in every step, people infect, each individual infects 2 people, so the reproductive number, the R0, would be around 2. Then, in parallel, once the first sequencing data became available, we, in our research group, asked, well, there's China, but there are all those other places where the virus started spreading. What's the R0 around the world? And at some stage, there was, well, maybe very optimistic hope that while Wuhan things got very quickly out of control, maybe climate, environmental factors are so different that in other countries, the spread would be slower. But what we found based on sequencing data, and in a second I'll outline how from sequences you actually can also get the R0, we got an R0 between 2 and 3, maybe even a bit bigger, consistently for the different countries. Meaning wild-type SARS-CoV-2 has an R0 between 2 and 3. So at least 50% of the population needs to be infected before the epidemic is starting to decline. And that already, knowing back then that the case fatality or the infection fatality was around 1%, it was clear that this will not, or there would be overburdening of the healthcare system if one would just let the virus run through. So how did we get this or calculate the R0 based on sequencing data? And here I should say again, this was not, that we developed at the time a new method. This was again based on decades of research by many different groups. So you have sequencing data here, hypothetical. I say we have 4 SARS-CoV-2 sequences from a particular country. They differ at particular positions just because in different infected individuals, the virus may have fixated a different nucleotide. Based on similarity between the sequences, we can reconstruct a phylogenetic tree. So that's the whole field of phylogenetics. And this phylogenetic tree, we can see as a proxy for the transmission chain. So we can approximately assume that, this patient here, or the virus from an infected host here, infected onwards yet another infected individual from which we sampled another sequence. So we have a tree, but of course, the tree doesn't help us yet to get the R0. Now we ask, how quickly actually do those transmissions happen? And if we had a sequence from every single infected individual, we could now just count through time how fast are the lineages accumulating. And if every 5 days there's a doubling, well, that would mean the reproductive number is 2. Of course, we only have a few sequences out of a big epidemic. So statistical methods need to now also incorporate that we have incomplete sampling, but the idea is essentially counting lineages through time, correcting for incomplete sampling. And the whole field of phyloDynamics is called phyloDynamics, extracting from a tree population dynamic parameters, such as the reproductive number. And you now may wonder why actually did we use genetic sequences and not just look at the case data? Well, for countries, say, like Italy, all of a sudden, all those cases appeared. And it was not clear if very early on, up on the first introduction of the virus, cases were actually already counted well. I mean, we were all short on PCR testing, etc. While with sequences, even if we sequence a couple of weeks later, we can reconstruct what happened right at the start of an outbreak. And that's why we use this alternative approach. And we're then rather convinced that an R0 between 2 and 3 is plausible, given from sequences we got the same as Rio and Althaus got for case data in Wuhan. Okay, so then we all realized around the globe that actually, well, this pathogen is spreading very fast. We are in the middle of a pandemic. So what we wanted is not to only know R0, so the reproductive number at the start of the pandemic, but also how it's developing. So different countries wanted to know then the R value, how big it is to see how well do measures work. One, say there's a mask mandate, by how much percentage does the R value drop? That allows us to consider how many measures we actually need to get the pandemic under control. And so we set out first for Switzerland, but then we downloaded data from around the world every day in real time to estimate the R value throughout the pandemic. And what was the core idea, or in detail, how did we calculate the R value? Well, so if at some day, say that was April, during the lockdown in Switzerland and most of our countries, we had, say, 700 new cases. And 5 days before, and remember 5 days, it's roughly how long it takes until one person infects its offspring, there were 1,000 cases. So we had an R value of around 0.7. However, not on that day when we saw 700 infected individuals. Roughly, it took 10 days at the time until a new infection got confirmed and reported in Switzerland. So actually, the reproductive number of 0.7 was 10 days earlier already. So we always have a bit of a delay quantifying our epidemic. So in essential, what I just described, how to calculate the reproductive number, is based on a renewal equation. There's a method implemented by Ann Corey already in 2013 at Imperial College, EPI-ESTIM. That was a method I remember reading this paper in 2013, thought it's a very nice R package, useful. I tried it out for some influenza data here and there. And then during the pandemic, it's fair to say around the globe, different countries, different researchers who were involved in the response used this package, where essentially the R calculation is based on the idea I just outlined, with the exception that, well, not exactly after 5 days are we transmitting, but there is a distribution. The WS is how infectious are you after S days. And so if it's exactly 5 days, then W5 is 1 and everything else is 0, but more realistically, you would have a distribution. And so essentially using such a renewal approach and of course having parameters around it matching COVID-19, we then reported R values throughout the pandemic. And now I want to highlight how the R value in 2 examples actually impacted public health policy making. That's based on how our R values were used. But here I really want to emphasize that I'm just showing now examples what we did, but around the globe, similar approaches by many different people were developed and used in their own countries for helping in the response. So in Switzerland, our Federal Office of Public Health got very interested in knowing the R value per day, which then actually led to that the government put this R value into a law at some stage, end of 2020. What they did was, so the Federal Office of Public Health every day reported this R value calculated based on what I just showed a slide ago. And then in some periods, pretty much when we were in the height of the second wave and then this new variant Alpha came, restaurant closures in Switzerland were actually ordered when the estimated R value was above 1, and then restaurants were allowed to open again when it was below 1. I guess it's something for later over a coffee to discuss how much sense that makes to have it that fast going back and forth. But anyways, to look at the positive side, it was a time when the politicians actually really wanted to have evidence for making their decisions. A year later, then, you may remember, there was the next variant of concern. It was Omicron. Most countries were in a Delta wave, and all of a sudden, South Africa, there was a briefing by the health ministry. They reported about Omicron. And as one puzzle piece to convince pretty much the world that Omicron is a real problem is they calculated, so they were very low case numbers, and then we always, in collaboration with them, updated also the R value for South Africa and their different provinces. It just jumped up, this R value, from well below 1 to roughly 1.5. And that was exactly when in the sequencing data, they all of a sudden saw a dominance of Omicron. So it was one puzzle piece here showing or quantifying how much faster Omicron can be transmitted compared to the until then dominant Delta variant. So those were now examples, 2 examples, and there would be many more by people around the world where the R value was actually used by policymakers to have evidence for decisions they want to make about the public health policy, be it tightening measures or having other interventions. Another concept which was used quite frequently was logistic growth. So just to get us all on the same page, we suppose a population X grows in size exponentially. So after time t, when we start with a size x naught at time 0, we have e to the beta being the birth rate minus mu being the death rate times t individuals. So classic exponential growth. And then suppose some other population Y grows in size according, again, exponential growth, but now the difference to the birth rate is a bit bigger than in population X. So it's beta times 1 plus this growth advantage F. And then we can ask, what is the proportion of Y in this total population X plus Y? And so if Y is first very small, well, it'll grow in size because it has a transmission or birth advantage. And in fact, one can easily take, the ratio and get this equation, which is the logistic growth equation. And here, just to remind everyone, that's how logistic growth looks. This population Y first being very small and in frequency, so close to 0%, and then it outgrows population X because it has a birth rate advantage. Again, now, what does that have to do with COVID-19? Well, let's look at the growth of alpha, this first variant of concern. And again, going back 3 years by now, up to December 2020, there was predominantly the SARS-CoV-2 wild type. So there was what pretty much started in Wuhan. And of course, this virus accumulated mutations, but it was not yet different in a sense that it became more transmissible or evaded immune response. And in December 2020, the variant Alpha was reported by the UK, who sequenced a lot at the time, to be spreading with a transmission advantage. And so this transmission advantage would be exactly the birth advantage we had in our logistic growth function, this FC. So the proportion growth of this variant Alpha is expected to grow logistically. And, the speed would be determined by this FC, which was not totally clear at the time how high this number would be. but it was clear it is bigger than 0. So then and that's the logistic growth. I mean, that's a very basic concept, but it became highly important. So in Switzerland, a couple days before Christmas, we got the phone call by our health minister in Switzerland, and he asked, well, when will alpha be dominant and cause a new variant wave? And he wanted to know, when do they have to do something? Because we were, mentioned before, in the middle of the second wave, and already then the hospitals were overwhelmed and couldn't cope with yet more cases due to a variant. Well, this was a few days before Christmas, and of course we can only know how alpha is growing if we know what's the frequency of alpha now, tomorrow, the day after, so we can fit a logistic growth curve to those frequencies. At least at ETH, normally between Christmas and New Year, all the labs have to be closed, even for security reasons. And so I called the ETH president, say we need to keep those genomic labs open. The government wants so. And so all went very smooth. It was one of those positive experiences that people just were very keen to support and understand the situation. And also the genomic, then the other call was to the genomic lab, if they could sequence, and those people actually were very happy we were anyways in lockdown, to have something to do. They couldn't travel anyways, anywhere to their families. So it was a very busy Christmas break in those genomic labs. And so a few hundred SARS-CoV-2 samples were sequenced in the week over Christmas. The first alpha sample in Switzerland was then detected on Christmas Day. That was actually a lab in Geneva. And then we determined, based on all the sequencing being done in our lab and other labs, what's the proportion of alpha among the sequences until the end of 2020, such that we know how fast is alpha spreading here. And so you see the case numbers here in the top in Switzerland, and that's end of December 2020. So we're in a stabilizing phase, so after this big second wave. And the proportion of alpha, this is just 2 weeks, we see going up from around 0.5% to 3% roughly. And from that, we fitted a logistic growth curve. So just the equation I showed you before, and predicted or projected that by March, alpha will be dominant. And if transmission happens at the time then, cause another wave. So in March, we will have another wave. And actually, that's how it turned out retrospectively. So indeed, it was dominant by March. What the government did, anticipating there will be a wave in March, that actually during dropping case numbers, namely early January, they tightened the measures. I think it was one of those moments where it was very hard to explain to the population what is being done. People thought they are out of the danger zone. Case numbers are dropping, while we saw this new variant underneath growing. And so the government did something. And so, yeah, again, there were more restrictions going through February to dampen the March wave. Okay, so with this, I want to come to the end of my talk. Where are we now? Where could we be heading into the future? And actually now, surveillance got downgraded a lot. Also in Switzerland, we barely sequence any clinical pathogen SARS-CoV-2 cases. However, nevertheless, in summer in Switzerland, there were quite a bit of headlines, and even Switzerland made it to the Washington Post with this new variant BA.2.86. And I want to just cover or give that as an example where we could be heading. So BA.2.86, as you may remember, was detected first in August. And the first 5 countries were the US, UK, Denmark, Israel, and South Africa, where in some patients this variant was found. And those countries still, in relative terms, much less than at the peak of the pandemic, but they still sequence, some fraction of their population. So why did Switzerland actually get into the Washington Post headlines? Well, actually, in Switzerland, a surveillance system was built up where wastewater was analyzed. And Switzerland was the first country to detect this new variant BA.2.86 in wastewater. And actually what is being done at those wastewater treatment plants there are samples. Every hour samples are taken, and once a day, everything is then mixed together. This is shown here is Zurich. So all of us living in Zurich, we contribute to this pond here. Then the wastewater samples are shipped to the lab, and actually not only from Zurich, but from 15 different places. The samples are prepared, and by now not only SARS-CoV-2 is considered, but also influenza and RSV, with the vision to expand further to other pathogens. And then with digital droplet PCR, the concentrations are measured, which is essentially a proxy for how many cases do we have at any point in time. And with sequencing, we can determine which variants are around. And so you get then those plots, how fast a variant outgrowing other variants. And pretty much with the models I showed earlier, so the reproductive number, the logistic growth, you can analyze this data. And then in August already indicated there will not be a major BA286 variant wave, but sure enough, cases probably will increase due to waning immunity and seasonality, what also turned out to be the fall wave. Here, pretty much all I covered, we are now using for wastewater, except for the phylogenetic trees. This is much more work ongoing because it's not so clear how to reconstruct the trees in wastewater. You only have short reads from different variants, but not a consensus sequence of one virus. So it's not as straightforward to build trees and do phylodynamics, but I think that's a very exciting research area for the future. Being at the future, what are our challenges and opportunities? Starting with the opportunities, I think throughout the pandemic, different countries showed that they can set up surveillance networks, gathering relevant data and by now also with all the new assays on a variety of pathogens in real time. I also think environmental sampling, such as wastewater, can serve as a great data source going forward, and that really got a push throughout the pandemic. And many models, also like the concept of the reproductive number, were developed further in the pandemic to enable very fast real-time estimation and robust estimations on huge data sets. And robust is also important if anything is used for policy making. So I think in a way, the pandemic accelerated that we're in really good shape, that we could do good surveillance and apply the models to the surveillance data to have insights, both for the point of surveillance, but also for understanding epidemiological dynamics in more detail. Challenges, of course, will countries actually invest in this monitoring of pathogens? How will the data be shared rapidly? And the people involved in the sequencing know probably about all those recent GISAID articles. And then last but not least, of course, can and will modelers be again so committed as they were in this past pandemic if a future crisis occurs, given all the backlashes many different researchers experienced. With that, I want to thank all the group who did do all this great work, all our funding, and thank you for the attention. I'm very excited to exchange now further in the question session. Thank you.